Oh, what do I have in here?
It's a brain!
Okay, so again, I'm not a zombie, but today we're going to be working on our mammalian brain dissection.
So brains are really cool, and there'll be a whole other video where we'll talk more about the structure and function of different parts of the brain, but today we're just going to review the dissection. So, and remember, every aspect of who we are, from our personalities to how we sleep, is going to be governed by the brain. And the brain is just part of our nervous system, and again, we'll explain more about the nervous system in other videos, but today we're just focusing on our mammalian brain structures and how we're going to do the dissection.
So the human brain has about 100 billion neurons. That's so many. But we're not actually dissecting a real human brain. We're going to look at another mammalian brain, which is the sheep brain. So all mammalian brains have a lot of similarities, and so we're going to talk about some of the things that are similar with our sheep brain and our human brain, and some of the differences as well, before we dive into our dissection.
First of all, the location of the cerebellum on the sheep brain is a little bit different from that of the human brain. Remember, the cerebellum controls muscle coordination, our sense of balance, and also our motor skill learning as well. It's more posterior on the sheep brain than it is on the human brain. Other differences in the sheep brain include where the spinal nerve attach, the pons, location of medulla oblongata, and of course the olfactory bulb is going to be a little bit larger, or comparatively larger on the sheep brain when compared with the human brain. Animals rely more on their senses and abilities of smell than humans do. You'll probably get a brain start out like this, and it's enclosed in this outer covering called our dura mater. And we can still kind of look at some structures before we actually take this brain apart. We can see the pituitary gland and optic chiasma if we look carefully. All right, so we have our pituitary gland in here. And remember, our pituitary gland is part of the endocrine system. It's going to produce critical hormones, which of course are chemical substances that are going to control lots of our body functions. And we also have our optic chiasma. Now, it's a little hard to see on this, this brain, but it's going to be where we have two optic nerves that are going to be crossing each other. And so that's visible before we remove our dura mater. Now, if we flip it over, we can see our cerebellum, which is back here. Of course, this dura mater is already a little bit removed, so that'll be in this location here. Okay. We can also start to see some of our gyri and sulci. Gyri are the bumps, and the sulci are the grooves, and these are going to make the classic squiggly pattern of the brain that we normally notice. But we're going to start to remove the dura mater, and the way you can do that is you can take your brain, and the tiny scissors will work well for this, and you can sort of poke a hole in any location and just sort of start a cut. And you can cut and slowly remove, being careful not to damage any of the tissue below. Make sure you guys can see. There we go. And then eventually, once you get large enough portions off, you can take a small piece and remove it. So let me cut this piece off. And then you can sort of pull up like this. And then you can start to reveal the brain so we can see the other structures a little bit better. Okay, we're going to continue removing the dura mater, and you guys will sit tight. All right, now sometimes if you're not careful, you can actually remove your cerebellum while you're taking off the dura mater because it's very loosely attached. So that's what's happened here, but that's okay. We still have all of these structures visible on the bottom. I'm going to actually continue working with this brain, and this one is still attached. So try hard to keep your cerebellum attached. It'll be easier to see some of the structures, but here we go. So if we look at our brain like this, we can see our superior and inferior colliculi, and we can also just barely see our pineal gland, which is going to be back in, oops. This pair is going to help us with optic reflexes and auditory reflexes in the brain. And the pineal gland, remember, which is inside here, is going to function as one of our endocrine glands that's going to help us secrete things like melatonin. Okay, so now we're going to take up our scalpel, and we're going to use our longitudinal fissure, so this line right here, as a guide, and we're going to separate the brain into our left and our right hemispheres. So be very careful where you're cutting this. You want to still be able to see the structures, and it's very helpful to use a sharp scalpel while you're doing this part. So here we go. Okay, so now we can see our arbor vitae in our tree of life inside our cerebellum, and this part between the cerebellum and cerebellum, again, is called the transverse fissure, so that's here. And remember, the cerebellum is only loosely connected, so if it falls off, that's hard. So here we have our lateral ventricle. We can actually stick our probe inside. And that's going to be sort of right next to our corpus callosum. And remember, that's going to link the left and right hemispheres in order to allow for communication. And we have our thalamus, which is here. That should be clearly visible. Things are medulla oblongata is here, our pons. We have our hypothalamus, which may be a little bit more difficult to visualize. Remember, that is going to regulate a lot of our body functions, like our heart rate, our body temperature, hormone levels, those kind of things. But as you go through, try to identify as many structures as possible according to your dissection guide. Okay, so the last thing that we're going to do as far as our brain dissection goes is going to be taking off a section of the brain to look at the difference between our gray and our white matter. So again, you want to take your scalpel, and I'm going to use this brain so it's a little bit easier to see. And you're just going to slice off a small part right here. And then once that is removed, we can really clearly see our gray and our white matter within this part of the brain, which will be interesting for you guys to visualize as well. Okay, so as you finish up your brain dissection, please make sure you are completing your dissection guide, you're identifying all the parts that you need to, and then when you're done, do not forget to make sure you follow our lab procedures for cleaning up. Okay, thanks so much for watching the brain dissection.